Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Pondicherry University M Sc Entrance exam question paper conducted in the year 2020 for the subject Biochemistry. So coming to the first question, what is the function of arteries in the blood? As we all know that arteries, they generally carry oxygenated blood and veins carry deoxygenated blood. And arteries carry oxygenated blood to deliver oxygen to organs, and veins carry deoxygenated blood back to the heart for reoxygenation. So the answer here is option 3, that is to carry only oxygenated blood away from the heart. So remember, arteries start with the alphabet A and away from the heart also starts with A. Away from the heart also starts with A. So arteries A. Away from the heart A. So when you see arteries, remember it is blood, oxygenated blood away from the heart. But the exception is for pulmonary arteries and pulmonary veins. That is, reverse is the correct one. Reverse is the correct answer for pulmonary arteries and pulmonary veins. Which organ is responsible for creating bile? So bile is produced by the liver and stored in the gallbladder. So the answer here is option 3, that is liver. Then next one. Adrenocorticotropic hormones will enter the bloodstream and act on which structure in the body? So they will act on the adrenal glands, specifically adrenal cortex. So the answer here is option 4, that is adrenal gland. Then which immune cell is part of acquired immunity? So the cells responsible for acquired immunity are lymphocytes, that is your T cells and B cells, dendritic cells, cytokines. And then you have the complement system also. So the answer here which is matching is plasma cell, because plasma cell is a type of B cell that secretes antibody. So answer is option A, 2. Then which structure releases calcium ions prior to muscle contraction? So it is sarcoplasmic reticulum. Answer is option A, 2. The next one. Which portion of the brain is responsible for controlling breathing and heart rate? So it is the brain stem that is responsible. It is beneath your cerebrum and in front of your cerebellum. It connects the brain and to the spinal cord, and it controls various autonomous functions, that is breathing, digestion, heart rate, blood pressure, etc. are some of the functions. Then next one, which cellular junction act as a barrier to prevent fluids from passing between cells? So between cells which act as a barrier is tight junctions. They don't allow fluids to pass between cells. Answer is 4. Then who is known as father of genetics? It is Gregor, Johann Mendel. Answer is A2. Then eukaryotic ribosomes are made up of dash and dash subunits. So they are made of 60 and 40S subunits. The 60S is the larger subunit and 40S is the smaller subunit. Then next one. Who discovered transposons in maize? So it was Barbara McClintock who discovered transposons in maize. Then which among the following is not a structural polysaccharide? So cellulose, it is structural. Glycogen, it is a storage polysaccharide. So answer will be glycogen. Pectin is also structural and chitin is also structural. Then next one. Which portion of the neuron receives electric signals from other neurons? So it is dendrites. Answer is A2. Then next one. If a cell is incapable of catalyzing very long chain, remember very long chain fatty acid chains, it is most likely to have a problem with which of the following organelles? So peroxisomes, they are responsible for beta oxidation of very long chain fatty acid, that is carbon atoms greater than 20. Whereas mitochondria, they oxidize mainly long chain fatty acid, that is C16 to C20. So the answer here is option 3, that is peroxisomes. So coming to the next question, which of the following will not result from enzymatic activity in a cell? Decreasing the activation energy of a reaction, that is because of enzymatic action. Then making a reaction more exothermic. That does not result from enzymatic activity. Increasing the forward rate of the reaction, that is correct. Increasing the reverse rate of a reaction, that is also correct. So the answer here is making a reaction more exothermic. Enzyme doesn't do like that. Make a reaction more exothermic. Then next one. What level of protein structure is determined only by hydrogen bonds? So you know about the secondary structure. There are two types, that is the alpha helix and the beta helix. And alpha helix is a coiled spiral and the helix is stabilized by hydrogen bonds between carboxyl acid of one amino acid and amino group of the next fourth amino acid. Then you have got the beta pleated sheets. In this also two or more chains are joined together by intermolecular hydrogen bonds. So what level of protein structure is determined only by hydrogen bonds is secondary structure. Answer is option 1. And next one. Which phase of mitosis involves the separation of chromatids? So it is anaphase. Answer is A4. What is the purpose of prostate glands in males? So to produce the fluid that nourishes and transports sperm or seminal fluid. So answer here is option 1, that is add to the fluidity of semen. Then next one. What term best describes when one species exhibit two or more defined phenotypes within the same population? So natural selection, you all know what it is. Polymorphism refers to multiple phenotypes that exist within a population, generally as a result of multiple alleles for the same gene. That is called as polymorphism. So answer here is option 2, that is polymorphism. What is allopatry? Allopatry, it is a mode of speciation that result in two separate populations because of geographical isolation, and this isolation prevent or interferes with the gene flow. Then assortative mating is a mating pattern and a form of sexual selection in which individuals with similar phenotypes or genotypes mate with one another more frequently than would be expected under a random mating pattern. That is assortative mating. So the answer here is polymorphism. Then which of the following proteins is necessary for nucleosome formation? So it is histones. Answer is option 1. Next one. Which of the following best describes an oncogene? A gene that regulates cell growth. A gene that stimulates apoptosis in cell. A gene that no longer makes a viable protein. A gene that causes uncontrollable growth. So an oncogene is a gene that has the potential to cause cancer. So oncogene is a gene that causes uncontrollable growth. Answer is option 4. Then next one. Which of the following pairs is interconverted in the process of mutarotation? So it is alpha D-glucopyranose and beta D-glucose. Okay. Oh, they have given alpha D-glucofuranose. No. It is option A, that is alpha D-glucose and beta D-glucose. Answer is option 1. Then, which statement is correct? Most enzymes are proteins. Most enzymes are proteins, but not all enzymes are protein because ribozyme. Ribozymes is an RNA molecule with a well-defined tertiary structure, and it act like an enzyme, catalyzing biochemical and metabolic reactions within a cell. So the most enzymes are protein is correct, but all enzymes are not protein. All enzymes are lipids, that is also not correct. All enzymes are hormones are proteins, no. So answer which statement is correct is option 1, that is most enzymes are proteins. Then next one. Which of the following act as a precursor of coenzyme A? So it is pantothenic acid that act as a precursor of coenzyme A. Then next one. ATP is required in the transport of water molecules, all molecules across a membrane, molecules to areas of lower concentration, molecules to areas of higher concentration. So what type of transport require ATP? It is active transport, and active transport is the movement of molecule across a cell membrane from a region of lower concentration to a region of higher concentration against a concentration gradient. So from here, from the options, it is molecules to areas of higher concentration. So from molecules from areas of lower concentration to molecules to areas of higher concentration. So answer is option 4. Then next one. The earliest cellular life forms appear to have been bacteria. Answer is option 4. Then next one. Which of the following gases is least likely to have existed in the early atmosphere of the Earth? So it is oxygen, which was not present in the early atmosphere of the Earth. Then next one. Darwin explained this theory of evolution in a book called On the Origin of Species. Answer is option 1. Then the ozone layer around the Earth helps in. So ozone layer, it is help in filtering UV radiation, especially UVB. Answer is option 2. Then immunological memory is manifested during adaptive immune response, non-specific immune response, innate immune response, all of these. So it is in the case of adaptive immune response, and immunological memory means the immune system can remember the antigen that has previously activated it, and when they again encounter this particular antigen, they can launch a more intense immune response, that is called as immunological memory. So it is manifested during adaptive immune responses. The next one. Which of the following is not an example of symbiosis? So symbiosis, lichens is an example. Algae and fungi are partners in lichens. Then mycorrhizae, roots of higher plants with fungus, I think. Okay, anyway, it is a symbiotic association. Then tapeworm and humans, it is a parasitic association. So that is not an example of symbiosis. Clownfish and sea anemones also symbiotic association. Then the term detrivore include decomposers, primary consumers, secondary consumers, autotrophs. So decomposers, they are classified as detrivores, scavengers and saprophytes. So the term detrivore, it include decomposers. The science of classification is called, it is called as taxonomy. Answer is option 2. Then next one. The development of vascular system in plants allowed water to move from roots to the leaves, carbohydrate to move from the leaves to the roots, a rigid structure that allow plants to grow taller. So vascular plants, they are able to grow higher than other plants due to the rigidity of the xylem cells which support the plant. So that is correct. Then carbohydrates to move from leaves to the root, that is also correct. And water to move from roots to the leaves, so that is also correct. So the answer here is option 4, that is all of these. Then the process of double fertilization is unique to, so it is unique to angiosperms, and in double fertilization one female gamete unites with two male gametes. One of the male gamete fertilizes the egg resulting in the formation of zygote, and the other unites with two polar nuclei for the formation of endosperm, and this is found in angiosperm. So answer is A1. Then next one. The body plan common to both insects and annelids is, so it is coelom. They both are coelomates. Answer is option 3. Next one. The infectious substance of prions is, so prions infectious substance is proteins. Answer is option 1. Then which component of immune system is crippled in AIDS? So HIV cripples the immune system and it will destroy the immune cells called as the CD4 T cells, which are usually activated during the early HIV infection by T1 type 1 interferons. Now CD4 T cells, they are also known as helper cells because they signal another type of T cell, that is CD8 plus T cells, to destroy HIV infected cells. So the answer here is helper T cells. The next one. Several enzymes are involved in DNA repair pathways. Which one of the following enzyme is also referred to as suicidal enzyme? So the answer here is methyltransferase. Explanation I will give in the next picture. Okay. So here if you see, it is a mechanism of direct repair. So direct repair mechanism does not replace the altered nucleotide, but instead changes them back to their original structures. That is what is happening in case of direct repair. And here if you see, this is a case of alkylation of guanine, that is methylation or alkylation of guanine, where a methyl group is added to guanine and it becomes what is called as O6 methyl guanine. And this guanine, if it undergoes replication, that is over here, you will see that it is pairing with thymine instead of cytosine, and one strand is normal and there is no mutation in that strand. And this mutated strand, if it undergoes replication again, you will see that it pairs with T and you will get a new gene product. Sorry, not a gene product. You will get a new combination of bases. That is, instead of GC pairing, you will get an AT pairing. So that is what is happening. Now what happens in direct repair is, here the methyl group of guanine will be removed and the normal guanine will be introduced. Now how does this happen? This is done by the action of an enzyme called as O6 methyl guanine methyl DNA methyl transferase. O6 methyl guanine DNA methyl transferase. That is the enzyme. And this protein, it catalyzes the transfer of the methyl group. That is over here, this picture. Transfer of methyl group of O6 methyl guanine to one of its own cysteine residue. So this is the enzyme. I have drawn only this portion. Only a small portion of the enzyme which is required for explanation. I have not drawn the full enzyme. It's not possible. So here what happens is, this methyl group from O6 methyl guanine will be transferred to one of its own cysteine residues. This is the cysteine SH residue of this particular enzyme, and the methyl group is transferred to this cysteine residue, thereby inactivating the enzyme. That is why it is called as suicidal enzyme. And this methyl transferase is not strictly an enzyme, because a single methyl transfer event permanently methylates the enzyme, making it inactive in the pathway. And after transfer of the methyl group to cysteine residue of the methyl transferase, guanine nucleotide is replaced. So it is a case of direct repair. Then next one. Glycolysis is the pathway used by cells to extract energy from glucose. Oxygen is not necessary for glycolysis, and the presence of oxygen can indirectly suppress glycolysis. This phenomenon is known as Pasteur effect. Now what is Pasteur effect? Is the effect of inhibiting oxygen on the fermentation process. Effect of inhibiting oxygen on the fermentation process. What is Warburg effect? Warburg effect is defined as an increase in the rate of glucose uptake and preferential production of lactate even in the presence of oxygen. That is Warburg effect. So glycolysis is the pathway used by cells to extract energy from glucose. Oxygen is not necessary for glycolysis, and the presence of oxygen can indirectly suppress glycolysis. So Pasteur effect states that the effect of inhibiting oxygen on the fermentation process. So the phenomenon here, it is known as Pasteur effect. Answer is option 1. Then the largest reserve of energy in humans is adipose tissue triacylglycerol. Then the mechanism of action of several anti-cancer drugs is by inhibition of DNA synthesis. Which one of the following diacetyl molecule impair purine biosynthesis? So it is methotrexate that inhibits the enzyme dihydrofolate reductase, thereby inhibiting synthesis of purines. Hence, methotrexate can be used as an anti-cancer agent. So it will inhibit DNA synthesis by inhibiting the enzyme dihydrofolate reductase, which is involved in purine biosynthesis. So answer here is option 3. Next one, a simple bacterial test for mutagenic carcinogens is Ames test. And Ames test, it is a widely used method that uses bacteria to test whether a given chemical compound can cause mutation in the DNA of an organism or whether the given chemical compound is a mutagen. And usually the strain of bacterium that is used is Salmonella typhimurium that carry mutation in the genes involved in histidine biosynthesis. So the answer here is Ames test. Then next one, the coenzyme not involved in hydrogen transfer is FH4. Answer is option D4. Then which of the following is not a fat soluble vitamin? D is fat soluble, C is not a fat soluble, it is water soluble, A and E are fat soluble vitamins. Then next one, the first pharmaceutical product of recombinant DNA technology approved for human use. It is humulin, nothing but human insulin. So the answer here is option 1. The bacterial communication phenomenon that uses secreted signal molecules to assess population density is termed as. It is termed as quorum sensing. Answer is option 3. Next one. The mordant used in gram staining is. It is iodine. Answer is option 3. Then phosphatidylserine, an important component of biological membrane, is located in. So it is located on the inner leaflet, but sometimes it flip-flops to outer leaflet under specific condition. So the answer here is option 4. Then next one. Dark grown seedlings display triple response when exposed to ethylene. Which of the following is not part of triple response? So decrease in epicotyle elongation, that is a part of triple response. Rapid unfolding and expansion of leaves, that is not a part of triple response. Thickening of shoots, yes, and horizontal growth of epicotyle. These three are part of triple response, whereas rapid unfolding and expansion of leaves is not a part of triple response. So answer is option 2. Then next one. Which of the following macromolecules is broken down by pepsin in the stomach? So it is proteins that are broken down by pepsin in the stomach. Now, if a person has A blood type, which of the following statement is true? The person cannot be given type O blood. O is a universal donor. So you cannot say that the person cannot be given type O blood. He or she can be given. Then next one, a person can be given type B blood. It is not possible to give type B blood to A blood group person. You can give either A or O blood. Then next one, the person makes A antibodies, not A but anti-B antibodies. So the answer here is option 4, that is the person makes B antibodies. Next one, dash are specialized cell junction that connect the cytoplasm of two cells and allow for the exchange of various ions and molecules. So they are gap junctions. Gap junctions allow the exchange of various ions and molecules. So answer is option 4. Then next one, hemoglobin is a protein that consists of 4 subunits, 2 copies of the alpha and 2 copies of the beta subunit. How many individual polypeptide chains are present in a fully folded molecule of hemoglobin? So it has got 4 chain polypeptide chains will be present. 2 alpha chains of 141 amino acids and 2 beta chains of 146 amino acid residues. A scientist has discovered a mutation that prevents cell from passing anaphase of mitosis. Of the following option, which is the most likely target of the mutation? Microtubule formation, myosin, proteins involved in chromosome condensation and actin synthesis. So actin and myosin, they are not directly involved in this portion of mitosis. They are very important during cytokinesis. So it cannot be myosin nor actin. Then comes the chromosome condensation. Chromosome condensation has already taken place, that is during the prophase stage, and so mutation cannot affect these proteins. So now if the cell cannot pass the anaphase stage, the cell is most likely having trouble with the separation of sister chromatids, and this process it is mediated by the attachment of kinetochore microtubule to the kinetochore on the sister chromatids. So there is problem with the anaphase stage, means there is a mutation that is affecting the formation of microtubules. So the answer here is option 1, that is microtubule formation. Then next one, which bacterial reproductive process does not involve any genetic recombination? So conjugation, yes. Transformation, yes. Binary fission, it is an asexual method of reproduction, so it doesn't involve any genetic recombination. So the answer is option 3. Then next one, which of the following is the main component in the fungal cell wall? So it is chitin, that is the main component in the fungal cell wall. The next one, which of the following is not a constituent of virus? So RNA is here, DNA is there, ribosome is not a constituent of virus, lipids are also present. Then human beings have diploid cells. This indicates that, so human beings have somatic cells that are diploid. This means that each cell has 2 copies of each of the 23 chromosomes, and it is obtained from that is one is obtained from father and the other from the mother. As a result, the karyotype of the human being, it will show 23 pairs of chromosomes, and there will be a total of 46 chromosomes. So diploid cell contain 2 non-identical copies of the same gene, and a diploid cell will contain 2 separate alleles for the same gene in the genome, represented as 2 homologous chromosomes. So human beings have diploid cell. This indicates that humans have homologous chromosome pair. Yes. Humans have 2 stages of cell division. No. Humans have 2 chromosomes in each cell nucleus. No. Humans have both sex chromosomes and somatic chromosome. No. So human beings have diploid cells. It indicates that humans have homologous chromosome pair. Then next one. Which of the following accurately describes the promoter? So it is a protein that attaches to DNA in order to create mRNA. Promoter is not a protein, it is a sequence of DNA to which proteins bind to initiate transcription. So first one is not correct. Then the binding site for DNA polymerase on DNA. It is not the DNA polymerase, it is RNA polymerase. The binding site for RNA polymerase on DNA. The attachment point for a ribosome before translation. Ribosome bind to the 5' cap on a eukaryotic mRNA. So 3 is also not correct. Now fourth one, a sequence of DNA used to signal the beginning point of transcription. That is correct. Answer is option 4. Then next one, which of the following blots is used to identify known RNA fragments? So northern blot is the answer, northern blotting. And northern blot, it is very similar to southern blot, except that it is used to identify RNA fragments, and southern blotting, it is used to identify DNA fragments. And western blot, it is used to identify protein subunits and fragments, and eastern blot is used to identify protein modifications such as glycosylation. Then next one, which of the following statement best describe the mechanism of RNA interference? Now RNA interference, it utilizes small RNA molecules, that is miRNA or siRNA, to target specific molecules of mRNA and thereby repress their translation or cleave them into short fragments. That is what is happening in RNAi. So globally interfere with translation by blocking all mRNA. No. Interfere with translation by targeting specific tRNA molecules. It is not tRNA, it is mRNA molecules. The next one. Interfere with translation by blocking a target mRNA. That is correct. The next one. Interfere with translation by targeting ribosomes. That is also not correct. So the answer is option 3. Then next one. When would it be appropriate to use extraction in order to separate compounds in a solution? So extraction is useful for separation when there is a mixture of compounds in a solution that have similar polarities but different solubilities. Okay. So when would it be appropriate to use extraction in order to separate compounds in a solution is when compounds have similar polarities but different solubilities. Answer is option 1. Next one. Which of the following compounds is generally translocated in the phloem? It is sucrose. Answer is 1. Then if you run a pentavalent IgM through SDS polyacrylamide gel electrophoresis, how many bands are you supposed to get by Western blotting using alkaline phosphatase conjugated secondary antibody? Okay, now IgM is reduced in the case of SDS polyacrylamide gel electrophoresis, and they have asked how many bands are you supposed to get by Western blotting using alkaline phosphatase conjugated secondary antibody. They are asking for the number of bands obtained by Western blotting, not by SDS polyacrylamide gel electrophoresis. Remember that. So if IgM is reduced properly, you can expect 3 bands corresponding to heavy, light and J chain. And detecting these bands on the Western blot depends on the specificity of the antibody that you are using. It can be either anti-J antibody or anti-light antibody or anti-H antibody. So you cannot use all the 3 antibodies at a time. You can use only one of the antibody for Western blot detection. So the number of bands that could be detected by Western blot is 1. So the answer is option 4. Whereas if it was by reduced to SDS polyacrylamide gel electrophoresis, then you will see 3 bands. Okay, then next one. Which type of mutation creates a premature stop codon in mRNA? It is nonsense mutation. Answer is option 3. Then which gland releases oxytocin and antidiuretic hormone? So it is the posterior pituitary gland. The antidiuretic hormone, it targets organ is kidney tubules, smooth muscles in arterioles. And oxytocin target organ is uterine smooth muscles and mammary glands. So the answer here is posterior pituitary gland. The next one. Which of the following products is produced in the highest quantity during the Krebs cycle? So for every 2 molecules of pyruvate, because pyruvate is obtained by splitting of glucose molecule in glycolysis, and glucose molecule, it has got 6 carbon atoms and pyruvate 3 carbon atoms. So 2 molecules of pyruvate will be produced from 1 molecule of glucose, and these 2 molecules of pyruvate, they will enter the Krebs cycle. So for every 2 molecules of pyruvate that enter the Krebs cycle, 6 molecules of NADH are generated, and 4 molecules of carbon dioxide, 2 molecules of FADH2 and 2 molecules of ATP or GTP are produced. So the answer here is 6 molecules of NADH are generated. So naturally option 1 is the correct answer, that is NADH. Then choose the mismatch. Bilateral symmetry, fish. That is correct. First triploblastic is flatworms. Free living flatworm is planaria. Radial symmetry is larvae of echinoderms. That is the mismatch, because it is not radial symmetry, they have bilateral symmetry. So the mismatch is option 4. Then next one. A scientist is working with a breed of dog and has noticed that two traits, ear length and color, behave in a normal dominant recessive hierarchies. Long ears are dominant to short ears and black coloration is dominant to yellow coloration. If he breeds a long-eared black dog with a short-eared yellow dog, what would be the resulting phenotypic ratios of the offspring? So, option 4 is the answer. I have given the explanation in the next slide. So, A is long ears and small a is short ears. Capital B is black and it is dominant, and small b is yellow color and it is recessive. So a cross is made between a long-eared black dog and a short-eared black dog. So these are the gametes that are obtained, and upon crossing AB to AB you will get capital A small a, capital B small b. Upon crossing this with this you will get the long yellow ones, and if you cross this one with this one you will get short black ones, and if you cross this with this you will get short yellow. So one long long-eared black, then 1:1 long yellow is to 1 short black is to 1 short yellow. So that is the answer. Option D is the answer. Then next one. Calvin cycle take place in the dash, that is in the stroma, and occurs. So option 1 stroma, and cytoplasm is not the answer. So 2 and 3 can be eliminated, and yes. Fourth one also stroma, whenever appropriate nutrients. No, only in during the absence of light. So 1 is correct. Then next one, a karyotype analysis of a sample revealed its karyotype formula to be 47XX plus 13. This result indicates, so it is a trisomy of the chromosome number 13, because instead of 46 you are getting 47 and then 2X chromosomes. So it is nothing but a trisomy of the 13th chromosome, and a trisomy of 13th chromosome is Part 2 syndrome. Answer is option 3. Then a type of cancer that begins in the cells of the immune system. It is leukemia. Answer is A, 2. Then the disease African sleeping sickness is transmitted by. So African sleeping sickness is transmitted by tsetse fly. Answer is option 3. In human males, testosterone is produced mainly by. They are mainly by the lady cells. Answer is 1. Then next one. Some mushrooms are poisonous and popularly referred to as death caps and destroying agents. Which one of the following is the deadliest? So it is Amanita verna. Answer is option 1. Then flower is a modified shoot or a branch. Which one of the following does not provide evidence in support of the above statement? Axis nature of the thalamus. Yes, that supports that flower is modified shoot or a branch. Then leaf-like arrangement of the floral members. Yes, just like in shoots have their own way of arrangement of the leaves. Similarly, floral members are also leaf-like arrangement as in leaves of the shoot. So it is a modified shoot or a branch. Then homology of floral and vegetative buds. That is also support that flower is a modified shoot or a branch. Then occurrence of mitosis in the floral tissues. Mitosis in plants occur only at the meristematic tissue, that is the tips of the roots and the ends of the shoots. Mitosis does not take place in the floral tissues. So it does not provide evidence that it is a modified shoot. So the answer is option 4. Then, anoxygenic photosynthetic bacteria possesses bacteriochlorophyll which absorbs light in the. So, anoxygenic photosynthesis is the phototrophic process where light energy is captured and converted to ATP without the production of oxygen. And they have asked bacteriochlorophyll absorbs light in the infrared region. Answer is option 2. Okay, coming to the next question. How many times can a 6 kb fragment be present in 3.9 micrograms of DNA containing 6 billion bases. Assume molecular weight of one base pair to be 650 Daltons. So 6 billion bases. So you have got, they have asked, okay, before that they have asked how many times can a 6 kb, that is 6000 base pair fragment, be present in DNA containing 6 billion bases. So all these like 3.9 micrograms of DNA and 650 Dalton, they are not required. So 6 into 10 raised to 9 bases is 6 billion bases and 6 kb is 6000 base pair fragment. So divide them, you will get 10 raised to 6 times. The answer is not given in the option, but this is the way you have to do it. So that is the answer, that is 10 raised to 6. Next one. Which of the following sequence is logically incorrect? So chordate hominidae man, that is correct. Angiosperm Poaceae rice, rice belongs to Poaceae and it is an angiosperm, that is also correct. Then Insecta Crustacea butterfly. Crustacea is and Insecta both are different. Insecta have 3 body segments, a head, thorax and abdomen, and Crustacea have only 2 body segments, that is head and thorax. Butterfly is an Insecta. So option 3, but it is not a Crustacea. So option 3 is the answer. Next one. In a population at Hardy-Weinberg equilibrium, the frequency of the recessive allele that causes a genetic disorder is 0.3. So Q is 0.3. What percentage of the population is expected to suffer from this condition? So you have to find Q square, that is 0.3 into 0.3, that is 0.09, that is 9% is the answer. Answer is option 1. Then next one. Deserts are arid ecosystems that occur near latitudes of 30 degrees north and south. Which one of the following can explain this geographic pattern? So the answer here is option 4, that is patterns in atmospheric circulations called Hadley cells that influence temperature and moisture content of air. That is the answer. Then next one. Chromatin has several levels of structure. The bead on a string structure of DNA wound around the nucleosome has a width of. So it is having a width of 11 nanometers. This is the bead and this is the DNA that is wound around it. So they have asked for the diameter of this particular structure, that is bead on a string structure of the DNA wound around the nucleosome. It has a width of this particular nucleosome or this particular structure is 11 nanometers. Then a polypeptide chain is made up of 101 amino acid residues. The polypeptide has 200 bonds about which rotation can occur. Assume that 3 orientations are possible about each of the bonds. Based on these assumptions, how many random coil conformations are possible for the polypeptide chain? So they have given that a polypeptide chain is made of 101 amino acid residues. Now number of amino acid residues do not count. So the polypeptide has 200 bonds about which rotation can occur. Okay, so they have given that there are 200 bonds about which rotation can occur. Assume that 3 orientation are possible about each of the bond. So each of the bond the bonds can rotate and they can give rise to 3 orientation about each of the bond. So each bond has 3 possible orientations. Then based on these assumptions, how many random coil conformations are possible for the polypeptide chain? So each bond has 3 possible orientation. 2 bonds will have 3 square, that is 9. 3 bonds will have 3 cube, that is 27. possible orientation, and 200 bonds will have 3 raised to 200 orientation. So answer is option 1. Then next one. The ions that mediate the upstroke of a typical neuronal action potential is, so it is sodium, because the rapid depolarization or the upstroke of the neuronal action potential occurs as a result of opening of the voltage gated sodium channels. So the answer here is option 1. Under normal physiological condition, the calcium pump of the smooth endoplasmic reticulum pumps calcium into the, so it pumps calcium into the lumen by hydrolyzing cytosolic ATP. Option 1 is correct. Next one. An enzyme catalyst decreases the difference in free energy between reactants and products. Enzymes do not affect Gibbs free energy of a reaction. So all 1, 3 and 4 options they have told on free energy, and that is wrong. So the correct answer is option 2, that is decreases the activation energy of a chemical reaction. That is what an enzyme does. Then next one. Which of the following experimental data revealed that DNA has regularly repeating 3-dimensional structure? So it is through fiber diffraction. Answer is option 1. Next one. Microplasma colonies have a characteristic appearance like that of. So they mycoplasmas, they do not have a cell wall. So they have a characteristic colony resembling a fried egg, because when a fried egg develop on solid agar media as a result of growth of the central portion of the colony in the agar, and surrounding this central portion there is a light area of only surface growth. So it will look like a fried egg appearance. Next one. When a flower can be divided into 2 equal and similar halves by only 1 vertical division, it is categorized as, it is called as zygomorphic. Then next one. Which dye is used in Bradford method? In Bradford method, it is Kumasi Brilliant Blue G250 is used. R250 is for SDS page. Then next one. Which of the following is not a physical method of transfection? Electroporation is a physical method. Particle bombardment is also physical method. Liposome mediated transfection is a chemical method. It is not a physical method. And microinjection is also a physical method. So which of the following is not a physical method of transfection is liposome mediated transfection. Then next one. One of the segments of the core promoter sequence of RNA polymerase II is the. So it is the Tata box. Answer is option 3. Then next one. Which of the cyclins have or has essential function in the S phase of the cell cycle? It is A type. Answer is option 1. Then Graves' disease is associated with. So Graves' disease is an autoimmune disorder that result in the overproduction of thyroid hormones. So insufficiency of thyroid hormone, no, excess of thyroid hormones. 2 is correct. Then during urine formation, the filtration of blood at the glomerulus is, so it is a pressure dependent physical process. Answer is option. Then next one. Molar absorption coefficient of phenylalanine is 200 per molar per centimeter at 257 nanometers. What is the concentration, that is in grams per liter, of this amino acid that will have an absorption value of 1 in a cell of 0.5 centimeter path length at 257 nanometers? So they have given molar absorption value of coefficient of phenylalanine. They have given the absorption value as 1. The path length is 0.5 centimeter. So you know the formula, A is equal to epsilon Cl, and from there you can calculate C. But C is given in molar, or that is 0.01 moles per liter. Now we know the molecular weight of phenylalanine, that is 165.1 Dalton or 165.19 grams per mole. So 165.19 gram is having 1 mole. So 0.01 mole will have 1.6519 grams. So the answer here is 1.659 grams per liter. So answer is option 3. Then next one. In a small village, a doctor overprescribed ciprofloxacin, an antibiotic, to his patient. The result was that majority of his patients had developed a new strain of bacteria that have become resistant to ciprofloxacin. What is the possible route that may have caused these bacteria to become antibiotic resistant? So first option is the bacteria developed a way to prevent the antibiotic from entering their cells. That is not correct. They are asking how the bacteria became resistant to the antibiotic. So the antibiotic lost its potency. No, the bacteria became resistant to antibiotic. It has not lost the potency. The bacteria learned to metabolize the antibiotic. No. The antibiotic resistant gene was passed on through the plasmid in a group of antibiotic resistant bacteria. That is more correct. So answer is option 4. Then next one. A researcher is performing PCR to amplify a sample of DNA. Unfortunately, he forgot to add the DNA primer prior to starting the experiment. Which of the following results is he most likely to observe? The reaction will work, but the product will contain many undesired mutations. The reaction will work, but at a significantly lower rate. The reaction will be completely unsuccessful. The reaction will work, but amplify a region that was not his target. So without primers, there is no way for DNA polymerase to function. So the reaction will be completely unsuccessful. Answer is option 3. Then which of the following is not true of nucleic acids? Only RNA has hydroxide group attached to the 2' carbon. Yes. Only DNA is read in the 5' 3' direction. Not only DNA, even mRNA is read in the 5' 3' direction. So that is not correct for nucleic acid. That's your answer. Then next one also we will see both DNA and RNA have nucleotides held together by phosphodiester bond. That's also correct. ATP and GTP are nucleic acid derivatives. That's also correct. So the answer is option 2. Only DNA is read in the 5' to 3' direction. So that is not the correct answer. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.